We're here at Munich Airport on our way to Bremen, so our flight is leaving in a few minutes. We are going there to a place where great science is done, which is the Jacobs University, to give our first ever Explorer Award to Professor Springer. So yeah, let's go! So we made it to Jacobs University. Now we have to find Professor Springer. He might know that we are coming, but let's see if we can surprise him. Hello?
Oh,
hi! Surprise!
Oh, wow.
For your excellent research, you were selected from a lot of people as the explorer of the quarter. So.
My name is Sebastian Springer. I work at the Jacobs University in Bremen in Germany, and I'm a professor of biochemistry and cell biology. The subject of our work are so-called MHC molecules. These are proteins that are present at the surface of every cell. They also present peptides, fragments of tumor-specific proteins, and it means the immune system can recognize tumors and it can be used to fight tumors. I feel proud to be able to embody this connection between basic research and application. We asked ourselves, how does the structure of the protein change when it loses its ligand? And we worked for years on this until we found that there's a particular soft spot that this protein has got. And then we asked what would happen if we clamp this with something called a disulfide bond. And then suddenly came the revelation that this could be used to help diagnosis and to ultimately help patients. When I heard about the Prometheus, and I think it was obvious that this is what we ought to be doing. Our old fluorimeter would allow us to run 3 samples at a time. You would need maybe 4 hours for temperature ramp, and you would need at least 2 ml of sample for each sample. And this, compared with the specifications of the Prometheus, it's obvious why this would have accelerated our work by 2 orders of magnitude.
The first time I used the Prometheus machine was actually in the beginning when I was working on refolding MHC class 1 proteins. And what I use it for now is that for the T-cell sorting that I work on, you have to bind T-cells to MHC class 1 molecules. So I have to purify and refold those proteins, and in order to use them I have to make sure that they're functional. So for this I use the Prometheus to check out the thermal stability of the protein and make sure it works the way that it should.
Another advantage is this machine is very fast, unlike the other instruments that we are using, because we used to work for the entire day, and probably at the end of the day we used to analyze like some 10 samples. But with this machine we can analyze up to 48 samples in 1 hour of time.
If you have an HIV patient, for example, it would be so useful to predict the kind of immune response that they are going to be able to make, which depends on their MHC alleles, as one would say. And to understand this would be fantastic. And to see maybe in my lifetime to be able to make an impact on 1 or 2 of these diseases that is actually visible, that certainly would be a dream.
What a great afternoon here at Jacobs University. It was great to meet the students here, to meet Professor Springer, to hear about their ideas, to feel the energy that they have here in their team. So it was really exciting. This was just the start of a longer relationship between NanoTemper and Jacobs University. So I'm really looking forward to the future.